We learn here how to prepare optimum suspension, and this is based on optimization of pharmaceutical formulations. So we'll use the same logic to find the optimum properties. So what is a suspension? So these are tiny solid particles which are floating into a liquid. So as you can see here, there will be some particles. If you look here on the figure on the left, those particles are suspended or floating more or less into the liquid, and that means that leads to dispersion. And this is often used. This strategy is used when the drug does not dissolve completely into water. And that's why you often see this warning: shake well before use, uh, because otherwise the particles can settle. And there are many pharmaceutical examples, such as Calpol, antacids, and calamine lotion. So the challenge we have here is to keep the particles evenly spread, and this is very important to ensure correct dosing of the medication. So the biggest problem with this formulation of suspensions is how quickly the particles will settle. So if you look here on the left side, larger particles will often settle quicker than smaller particles. On the right side here, you look at the time for the settling velocity, and this is basically plotted against the particle diameter. And you can see this is going exponentially, basically smaller values, and it gets bigger and bigger as the velocity gets bigger and bigger as the particle size is getting bigger. So the secret or the way to control and produce a good suspension is to control flocculation. And as you can see here is a comparison between deflocculated particles in a suspension. So when this happens, the particles repel each other, and at one point they will just sediment and form a hard cake at the bottom, and that means they will be very hard to redisperse. On the opposite can happen is flocculated particles, and in this case they will just clump or come together loosely. As you can see here, they will form fluffy sediment and easy to shake, um, and then just come back. The aim is when we form a suspension is we want flocculation to be controlled, or just to make sure we get floccules, and therefore that will help producing a good suspension. Now the idea is to form this kind of suspension is we use the same of factorial design. So we want when we have a balance between flocculation and deflocculation, as you can see here again, is to have a balance between the sedimentation rates. So floccules will sediment fast compared to the deflocculated ones, and then on the contrary, floccules often they will be larger compared to the defloccules. When there's deflocculated particles, they will form smaller aggregates or smaller particles. They will be loose in terms of when you have floccules. Well, they'll be hard cakes. And then also redispersibility will be different, and stability, which is the main thing, is going to be different as well. For floccules it's going to be good, while for deflocculated particles it will often be bad. So how to prepare suspensions? As mentioned, the process of making a suspension starts with wetting the powder, and this is aided by the use of surfactants. And surfactants, they will have hydrophilic and hydrophobic part. And as you can see here in this animation, you will add a surfactant layer on the surface of the suspended particles, and that will basically help with coating the powder or the particles and help them to flocculate. So this helps, it makes the dispersion in water layer in water media much easier, and also it will prevent aggregation of the powder. Sometimes, or in many cases, depends on the type of the surfactant and the powder, we might need to adjust the pH. And in some cases, you might need to add some preservatives because that will help with microbial growth. But the pH is important, especially if you're using ionic surfactants. A very important concept in understanding the stability and the formation of suspensions is zeta potential. So particles, when are suspended, they will have an electrical charge. And the way we measure this electrical charge is called zeta potential. And as you can see here in this diagram, you get the particle and it will have a charge on the surface. You will have some bound layer of particles, of charges, and then you'll have something called diffuse layer. And the charge across these two layers is what really constitute the zeta potential, as you can see it here highlighted. And if you take this this kind of circles and you express it in a two-dimensional way, and assess the stability on the y-axis and the zeta potential, so you'll see that you can end up with a kind of optimum values, and you will see that the minimum stability is when it's around close to neutral. That makes sense because you will particles will basically aggregate, and they will have excellent stability when there is some charge on them. So as mentioned before, this is good to some extent, because if they have too much repulsion, you end up with deflocculation, and then maybe you end up with formation of cake and hard to redisperse, so deflocculated system or particles. So the aim is to have some high value, but not high to the extent where they can just end up with being deflocculated. On the other hand, there will be some kind of attraction, but they will end up with flocculation. You need to have a balance between both properties. And the one thing we can do here is to add some electrolytes, because electrolytes are basically salts, and the opposite charge will adhere or migrate to the surface of the particle, and that will help neutralize the surface or affect the zeta potential of the surface, and that hopefully that will give you the right kind of balance between the properties of the suspension. When preparing suspensions, we can choose dependent variables that can affect the quality of the suspensions. And here we are referring to the factorial design. And X1, in this case, we decided to vary the surfactant concentration. And we said the surfactant is important because it helps to wet the dispersion or the suspension. So we choose three ranges from 0.1, 0.5 and 1%. On the other hand, we now understand that the charge at the surface is important, and therefore we can vary the electrolyte concentration. And we likewise here we use three different ratios as shown here. The rationale here is because too much electrolytes, if you add too much of your electrolyte or the salt, then that can cause deflocculation, and that means you get repulsion, and eventually hard cake is forming. One common independent variable that we use to assess the quality of our suspension is the use of sedimentation volume. And as you can see here in this animation, we start with we have our suspension we do a number of shakes just to ensure that we have uniform way of redispersing the suspension, and that way we can compare different batches similarly. And then after that, we allow the suspension to settle down, and we measure the volume that is formed after the suspension has completely settled. These are the tests that we can use to assess the quality of the foam suspensions. The first one, which we already explained, is the sedimentation volume. And that means basically you allow the suspension to settle and you measure the volume. Of course, the higher the volume is, the better, because that means there's less likely forming cakes, deflocculated system, and there's more like flocculated, so they're loose and they can be easily to resuspend. This is related to the redispersibility, and in this test is you count how many times you need to shake the mixture to redisperse. And of course, the fewer, that means is the better suspension. The third one is the dose uniformity. Basically, here you sample from top, middle, and the bottom, and you measure how much of the active in each area region. And the closer, the lower the variance, that indicates a better suspension. That basically there is a more uniformity across the whole suspension. The fourth one is the viscosity, because that will affect uniformity and how easy to pour the suspension. You don't want it to be very watery, very thin. Finally, it needs to be stable. The pH should remain the same, because you don't want to have some shifts in the pH. In terms of expected results and interactions, these are what you would expect. So you look at the contour plot, your map, and you would expect that if you increase the surfactant, then you should end up with a better wetting, and therefore you would expect that you end up with a uniform suspension. If you increase the electrolyte, then there is more flocculation, but then more flocculation means uh, better remixing, because you don't end up with the deflocculated system. But if you keep adding more of your electrolytes, then you end up with deflocculation, and that will have the opposite effect. When you look at the interaction, we always talked about interactions. The surfactant affects how electrolyte works, so there will be an interaction between both of them. And the reason for this, because in many cases the surfactant will be a salt of a fatty acid. So for example, sodium lauryl sulfate is an example. Even if the surfactant is not ionic, it will interact by hydrogen bonding. So in many cases, many of the pegylated, ethoxylated uh, uh, surfactants, they interact with water through hydrogen bonds. And that hydrogen bonds might be shift when you have the electrolytes in the system. So anyway, that means you need to look at that effect more carefully. And the easiest way is, once you put all your data, you build your contour plot, and then you look at the map, and that gives you the optimum region.